So first of all, thank you very much for the invitation. And the first thing I want to mention is that this is not exactly personalized medicine, what I'm going to speak today. This is kind of making a step back. So here we are going to try to understand the underlying biology, which in the future might help us to produce novel targets, for example, for personalized medicine. But the technology, as Chris mentioned in the morning, single-cell sequencing is still quite expensive to do it in a whole tumor sample, for example. So in few years to come, we'll see that this might be able to be used exactly for patient-specific. Nowadays, it's more about understanding the underlying biology. So a little bit of an overview of what I'm going to talk today. Well, I think you have heard a lot about single-cell transcriptomics, especially yesterday Oliver. He spoke about their approach to single-cell transcriptomics. But I want to give a little bit of an overview what of the experiment and kind of like a simplified version of what is going on behind, what is the actual data that we are getting to work with. Then I'm going to talk a little bit about which are the challenges that are proper of the single-cell data as opposite of previous RNA-seq datasets where we were sequencing basically lots and lots of cells all together. So there are some things that didn't appear, like noise, because we have so many cells together, like expression was averaged out in a way, so we were reducing the noise. And then we will talk about our approach for modeling these issues, and finally, how we fit these models. I'm not going to talk during this presentation about the actual application, because that will be left for the tutorial session this afternoon. So single-cell transcriptomics. So biological heterogeneity. Well, there are many layers of biological heterogeneity. We can think that we can have different species, but also within one specific species, we will have a very wide range of phenotypes or differences across the subjects in that species. But also, we will have, if you look internally, we will find different organs, and each of these organs will have a different function and different shape. And then again, if we look within each of these organs, for example, in the brain, we'll have that it's a very complex structure, and some cells will have some function, and some other cells will have a different function. So we are every time going more and more deeply trying to understand what is going on internally. So most of the transcriptomic studies that have been published up to a few years ago, they were only focusing on overall expression across these big bulk samples of cells. Nowadays, it's possible to sequence or to study gene expression at the single-cell level. And that will report us about a whole new layer of biological processes. For example, in tumor samples, it is known that cells are very, very heterogeneous. So we will not have only one cell type, we will have multiple cell types that are interacting. And only single-cell sequencing will allow us to disentangle this heterogeneity. So a little bit of a very simplified version of what is the experiment. So basically, we will start with a sample, with a tissue sample, and then what will be done is to dissociate and isolate particular cells. So we will separate each of the cells. Here we have only two, just for illustration processes. And then we'll look inside these cells, and we will extract the RNA from the nuclei. Once the RNA is extracted, they will synthesize cDNA, which is a more stable molecule, and we can actually sequence this molecule. Afterwards, what happens is that because we are working with single cell, the genetic material, the mRNA content is very small. I mean, I've seen, for example, worms which have like 1,000 cells, and you really need to look at the microscope to look at them. So one cell is really, really, really tiny. So what is done is to amplify the molecules in such a way that we have enough material to be able to sequence and to learn about it. Once the material is amplified, what will be done is to sequence, so basically read each of these molecules and map them back to a reference genome. Once we do that, we will say, for example, this molecule here, in this case, is mapping to gene 1. So we have 3 copies. We have one original molecule, but because we amplified, we have 3 now, and then we will have this measure for this gene at this cell. And as Magnus was saying before, because of all these amplification stages, we don't really have like an exact number of the expression. We only have relative changes on expression because we don't really know the absolute number. So finally, the data set that we will work with, or what is the output that we will deal with it, is like a big table. So where we will have all our genes and all cells, and we'll have this kind of count data, where we will count in every single of the cells and the genes how many molecules of these molecules that we sequence are mapping back or look like each of these particular genes. So what is the power of single-cell RNA-seq? Well, the power is to disentangle this heterogeneity that we have within tissues. So for example, using this single-cell RNA-seq, we will be able to distinguish novel cell types that can be potentially targets for new medical treatment. So as a notation, I'm a statistician, so I like to think about this, how the data set looks like. And for me, my data will be this big matrix of counts, as we spoke before. And each of these elements will be, I will only index first the gene, and second the cell. And then this will be my count data, so the number of mRNA molecules in each cell that are mapping to each gene. And just to have a snapshot of how real data looks like, so for example, these are only like the first 10 genes that I had and the first 10 cells. And you can see that there are lots of zeros, first of all. These ones were RNA spikes. I will explain you later on what are these. But these are artificial molecules that are there. We know that they are there. We added them. And you can see that still we see quite a lot of zeros. And this is because we have very small amount of material, so it's very easy to lose things. We cannot really capture them. And the other thing that you can notice is that even though these molecules were added exactly in the same quantity to all the cells, I get quite a lot of variability in these numbers. Okay? So these are some of the challenges that we will have. So going to the challenge. So the first of them is the normalization. I think this is quite well known that when we have, not only in single-cell RNA-seq, in many types of data, we really need to normalize our data in order to put everything in a comparative scale. So we want to somehow get rid of variation that is unwanted. For example, one source of changes on the scale is this amplification step that I showed you before. So if we come back to this diagram here, you can see that even though these two cells had 3 molecules each at the beginning, after amplification, I ended up with more molecules here than in the other one. But that's not any biological difference. It's just that one of the cells was amplified more than the other one. Yeah? So once we have the data, we really need to discount this kind of amplification that happened there to put everything in the same scale. So here it was an example with, imagine the same thing. So we have this cell here. We had these 6 molecules, and then the other cell was amplified, and everything was counted twice. So if we get counts from this cell here, we will observe that all of them will be roughly twice as much as the counts on the first cell. Another source of changes on the scale is the total mRNA content of the cell. So if we have a cell that has more mRNA content in total, we will expect to have more counts than in a cell that has less mRNA content. So we want to draw these things to the same scale. So normally, what the pipelines will be to take the raw data and to normalize it as a first step. So one very common way to normalize it is to calculate the RPM normalization, which basically will take the total number of counts that I have in a cell and then divide the actual counts by this proportion. And they are multiplied by 1,000,000 as well. However, there has been some people showing that this kind of normalization is can be very affected if you have some kind of outliers or like some very rare cell. So therefore, a few years ago, Anders and Hoover, they developed this Deistic method in which they have a different type of normalization. So they basically, the idea is to take the counts and to look at them as in reference to like an overall cell. But instead of taking the total number of counts, they take a geometric mean. Okay? and they show that this is more stable to outlying observations. Now, issues. These strategies perform quite well for bulk data sets where we didn't have all these zeros and technical variability was much smaller. However, for single-cell RNA-seq, you can see that they are quite noisy. One of the reasons, for example, for the Deistic factors is that we have this geometric mean here. So if we have one gene that has 1 0 in one cell, we cannot use that gene for the normalization because this term here will be 0, the product will be 0, and then the division will be undefined. So what we will have to do is to exclude from the normalization step all those genes that have at least 1 0 and calculate our size factors. The issue with single-cell RNA-seq is that because lots of genes will have lots of zeros, we might end up calculating our normalizing factors based on only a few genes. Okay? The second challenge when analyzing single-cell RNA-seq data is technical variability. So, for example, there was a dilution series that was prepared in Brennecke et al. Paper a few years ago. So basically, they look at 2 technical replicates, so 2 identical cells, and they sequence, and they try to compare the expression between these 2 technical replicates. Okay? And then they started diluting the material. So at the end, they started with a big amount of mRNA, and then they started reducing the amount of mRNA in order to look more like how much mRNA we have from a cell. So if we have a lot of mRNA, and we look at 2 technical replicates, these are the read counts in the 2 cells across all the genes, you can see that they are quite strongly correlated. So this is more or less like a diagonal line. However, if we start reducing the amount of mRNA, this starts diverging from the diagonal. So we have noise. Okay? We have a gene that is, we know that is expressed in the 2 cells, but here, for example, in this cell is lost, whereas in the other cell we capture it. Now, if we want to look how much mRNA content we have in a cell, for a single cell, we are kind of in this situation. So you can see that there is quite a lot of noise in single-cell RNA-seq. And if we don't take that into account, we will not be able to extract biological signal. So we need to quantify this technical noise. What can we do? In general, to quantify some noise, we need some kind of control. So what has been designed so far is to use non-biological spiking genes that are not expressed on the cells that you are studying, but you add them artificially. And you add them in a known quantity. So you know, for example, I have 10 mRNA molecules here, and in every of my single cells, I have 10 mRNA molecules of this gene. Now, because we know this, that will give us like some kind of gold standard to which we can compare our empirical measurements. So if I knew that I have 10 and I only capture 5, I'm losing those 5. So normally, the most typical set of molecules that is used is these ERCC molecules, which were these 92 extrinsic molecules that were designed in order to cover a wide range of spectrum. So we'll have some that are very lowly expressed, very lowly concentrated, and some that are more highly expressed. So if we, in the same paper, they look at an experiment just looking at these spiking genes. They didn't use the ERCCs. They used another kind of spiking genes where they had much more. And they look at the expression of these spiking genes in 2 technical replicates, 2 identical cells. And you can see that there is some kind of noise. So here we have one cell, here we have the other cell, the read counts, and they are not a perfect line. There is some sort of correlation, but especially in the genes that are in the low end of expression, there is quite noisy. Now, if we compare these, which are only technical genes that we add in the same concentration, to biological genes that where we have some kind of biological variability, we can see that this is less correlated than this. So the difference, sort of speaking, the difference between what we have here and the correlation that we have here will be some indication of what is the biological noise. So we want to, from this picture, kind of extract or tease out what is technical from what is biological. So this is what has been done in that paper. So basically, they look at the so these spiking genes, and they computed the coefficient of variation that they took for these spiking genes, and then the average expression rate. And then they looked that there was some kind of relationship here. And they fitted this relationship, so they fitted this curve. Once this curve was fitted, what they did was to look at the biological genes and overimpose this curve, and to say, well, everything that is above what we will expect as a noise from the spiking data, we'll call it biological variability. And for example, these genes here, they have a substantially higher coefficient of variation than what will be predicted just due to noise, and therefore they will call these as highly variable genes. Now, what's the issue of this approach? Well, this is a two-stage approach. So we are taking some data set, doing some fit, taking a point estimate of that fit, and then using that in a second data set for the other genes. So we are ignoring all the uncertainty that we have in the first fit. We are just using a point estimate. Lastly, but no less important, and this is quite critical, and this is something that you will spend, if you analyze single-cell RNA-seq, probably you will spend quite a big part of your time doing this part, is the quality control of your data. So while doing the sequencing, it's very often we will have some cells that were stressed during this process. So basically, the cell was kind of dead, and we don't really extract any mRNA from that. So we need to look at some indicators of whether this cell was damaged or not. Some of the typical indicators that we look at is the fraction of map reads. So when we map these reads back, there were some reads that will not be mapping back to any of the genes in the reference genome. So we don't really know what are these reads. And then, for example, if we have in a cell too many reads that are mapping back to the spiking genes with respect to the biological genes, that means that we don't really have any information about the biological genes, or something happened. Or we can have a lot of mitochondrial genes, so we didn't really have much information from the nuclei. There are other things that we can use, like experimental information. For example, they will look at the cells in the microscopy and see if they are like dead or not, and so on. However, we need to be very careful about not removing, for example, very rare subcell populations that might appear as an outlier, but is actually a biological meaningful sample. So let's look at our approach. So we will continue to work in a Bayesian approach, as has been done all morning today. So this is our model. It's called Basics, and this stands for Bayesian analysis of single-cell sequencing data. So what is Basics? Basics will be an integrated Bayesian hierarchical model in which we will try to deal with these issues that before were done in stepwise approaches simultaneously. So we will treat the normalization as if they were like model parameters, basically, and we'll estimate them together with all the rest of model parameters. Then we will use the spiking genes to calibrate the estimate of the technical variability, but again, not doing it in two steps. We will combine it in one step. So we will borrow information from all the spiking genes and from the biological genes in order to get this estimate. And finally, we will go for some downstream analysis. In particular, today I will focus on detecting or classifying genes that have a very high variability within the population of cells that we are analyzing. So for example, these are candidates to be markers of cell populations, or very lowly variable genes that look more like kind of housekeeping genes that are very stable and very highly expressed across all the cells. So let's build a model. So I will go step by step on how we thought and during while we were building this model. So imagine first that we have only spiking genes. So we have genes that we know are exactly in the same quantity in every single cell. And imagine that we have two completely identical cells and that the amplification was exactly the same. They have exactly the same mRNA content. Okay? So you can think sequencing as a sampling process. So from all the molecules that I have there, I will start sampling molecules and mapping them back to a particular gene. if there is no any difference on sequence on amplification or on anything, basically the rate of the expression at the counts that we will observe will only depend on the amount of mRNA from that gene that we have in that cell. Okay? And also, because we are assuming this Poisson counting process, we will have that the expected value and the variance will coincide. Now, if we go to a situation in which we actually had cells in which the amplification step was different in different cells, which is actually what we have in reality, we'll observe that the rate of the counts will depend also in something that is cell specifics. So this, for example, in this case, we'll think that this Sj will be 2 for the cell 1, for example, and 1 for cell 2. So just to say that here we will expect twice as much counts as in the other cell. But again, we observe under this model, we will have that the expected value and the variance will coincide. However, in practice, if we just take empirical estimates of normalized counts and we look at the expected values and the variance, we observe that the variance is much higher than what we will expect with a Poisson model. And that is a sign of the noise that we have. So the most easier way to include this over-dispersion of this noise in this model is to use a negative binomial model. And this is just a very complicated way to describe a negative binomial model in which we will have a random effect. So instead of before having the Sj, which was like a fixed effect, a parameter, now we'll have a random effect. We'll have a random quantity to which we put a distribution. And then that distribution will generate a negative binomial model. And in this negative binomial model, you can see that the expected value and the variance, they don't coincide. The variance is higher than the expected value, and the difference depends on this parameter theta. Okay? So if we don't really have any over-dispersion, we'll go back to the binomial, to the Poisson sampling. If we have over-dispersion, we will be in this case. So if we were working with for the spiking genes, more like bulk RNA-seq, this parameter, this distribution will look more like a Poisson than a negative binomial. So now what happens with the biological genes? All of this was for the spiking genes. Now we need to look at the genes that we are actually interested on. So we'll keep underlying the same structure that we had before. I just copied what I had here. However, we will have some extra things that we need to add in order to account what is going on with the biological genes. First of all, we need to take into account a second set of normalizing constants. And this is due because for the spiking genes, it doesn't matter the mRNA content that I had on the cell originally, I added exactly the same quantity. So all what I need to normalize is kind of these amplification biases. However, for the biological genes, if I have more mRNA content, I will have more mRNA molecules from that gene. So I need to normalize regarding to that mRNA content, or cell size, some people call it. So this will be this extra set of normalizing constants, phi j. Now, the second part is that, well, for the spiking genes, we knew that we put them exactly at the same quantity, so all the variability is just technical. Now, for the biological genes, we actually have some biological variability going on. So we have some genes that will be on in some cells, off in the other cells, or that it can be on in both cells, but just the level will be different level of expression. And this is what we are doing by having this parameter here. So this is kind of like an interaction term, you can think. So it depends on the gene, I, and on the cell, J. However, this is quite a lot of parameters, so we parameterize this in a hierarchical way, in such a way that the actual parameter is the variance of this distribution. So when we look at the expected values and the variance, we'll have for the expected value is very similar to what we have for the ERCCs, except that now we have this extra set of parameters. So here we had only Sj and μi, whereas here we have an extra normalizing constant. So we have that the expected value of the expression counts will depend on something that is gene-specific, so the concentration that was actually on the cell, also something that depends on the amplification, how much was amplified, the genes on a specific cell, but also the mRNA content in that cell. And now for the variability, we will have the first term that was exactly as before, but then we have an extra term here, which is controlled by this parameter delta I. So basically, if the gene was very like housekeeping-like, so if it was very stable, the variance of these parameters here will be equal to 0, so this term will disappear. So the model is quite complex, but this is like a graphical representation of the model. So basically, we have the counts. So imagine here we have 2 cells, and then we have one gene that is biological and one gene that is spiking, and we have the counts so for observations here. So we are borrowing information across all cells in order to estimate parameters that are gene-specific, and then we are, which in this case will be these mu i's that are representing the overall expression level of the gene, and these delta i's that control the variability of the gene. And then we will have some, we will borrow information across all the genes in order to estimate things that are cell-specific, which are these normalizing constants and the technical variability parameters. One thing that I need to add is that for this parameter, for this model to actually be able to learn the noise, we are assuming that we know the input number of molecules. So that's why this is a square and not a circle like here. Okay? So, well, again, this is quite a complex model with lots of parameters. Can we really estimate this model? A first question that I always get is like, well, you have 2 things that depends on J here. Sorry. You have 2 things that depend on J here, and then they are multiplied. Can you really tease those out apart? So to formalize this will be called identifiability. So what is identifiability? We need to check whether different parameter values will give us the same likelihood or not. So this is a very toy example. So imagine we have a normal distribution where the mean, we describe it as the sum between 2 different parameters, can be the multiplication as well. However, if we change the values of these 2 by adding to one constant and taking out that constant from the other parameter, we'll have exactly the same distribution. So from the data, there is no way to learn whether the true underlying values are alpha and beta or alpha and beta star. Okay? So we need to have some extra constraint to actually know what are the actual values. So what are our identifiability restrictions? Well, the first identifiability restriction that I also told you before is that we assume that the input number of molecules for the spiking genes are known for experimental information, from experimental information. By doing that, we can identify the scale of the SEA. So if we look at here, we have this expected value that was a product of 2 quantities. So the product is not, so the product will be identified, but each of these quantities by itself, there will be a scaling issue. So I can multiply all the mu i's times 4, for example, and divide all the SEAs times 4, and I will get exactly the same distribution afterwards. So by fixing those values, we fix the scale of the SEAs. So we can actually estimate these parameters here. Now, because all these 2 parameters are identified, we can identify this extra parameter theta. Now, for the biological genes, the issue is that we don't really know these quantities. This is something that we want to estimate. So what will happen is that we will not be able to identify simultaneously the mu i's and the phi j's because there is a scaling issue. The SEAs we can because the model is built simultaneously. So the SEAs, we can identify them from the spiking genes, but there is a scaling issue between this one and this one. As an identifiability restriction, what we impose here is to fix the scale of the phi j's. So basically, we just say that on average, they are equal to a fixed value. In particular, we use equal to 1. the value is quite arbitrary, but as I said, as Magnus said before, gene expression is only measuring relative changes, so the absolute number itself doesn't matter. Okay? So by using all these things, as we saw before, we can decompose the total variability for the biological genes in 3 terms. So we'll have a first term that we call it like baseline or chop noise, which is the noise that we will expect even if we have a completely homogeneous population of cells, nothing changed. Just because sequencing is a random process where we are sampling, we will have some variance there. A second term, which will be controlled by this parameter theta, is what we call technical variability. So this will only appear because we have this kind of different scale on the different cells. And then a third term that will be controlled by this delta I, which is the biological noise, or in a way, the true underlying biological variability that we have. What can we do with this decomposition? One thing that we can do, and Ollie spoke a little bit yesterday about this, is to quantify how much is the noise that we have. So for example, we can take from the 100% of total variability overall across all genes and cells, 20% was due to a technical component. And that can be informative. For example, if we have 2 different chips, we can compare the technical variability. This experiment was more noisy than the other experiment. A second thing that we can do is to rank the genes from those who vary the most to those who vary the least in terms of the biological heterogeneity. So what's important of this? So if we have genes that vary quite a lot, so they are very highly variable, like they are on in some cells, very off in other cells, these genes are very like drivers of the heterogeneity within the populations of cells that we are analyzing. And therefore, they might be markers or potential markers for novel subpopulations of cells. Now, if we are looking at lowly variable genes, we will recapitulate those genes that are related to very core processes of the cell. So they are very highly expressed in all the cells. And they also, it's important to identify this because they can reduce dimensionality in downstream analysis. So at the beginning, I told you that one of the applications of single-cell RNA-seq is to kind of characterize this by clustering these distinct subpopulations of cells. If we have genes that are quite stable across all the cells, those genes will not add anything to the cluster. So we can just remove them beforehand. So again, I told you I will look into detail this during the afternoon in the practical session, but this is more or less the output that we can get. So we can have an estimate, for example, of the expression rate of each gene on overall across all cells, and then some kind of estimate of the biological variability parameter, this delta I. In this case, we had a very homogeneous population of cells. These were mouse embryonic stem cells, and then we can see that on the high end of expression, so very highly expressed genes tend to vary less, so the delta I is very small. And in this case, very lowly expressed genes were more like related to genes that were varying more. And it's because the overall the expression is very low because it was just on in some cells and low in other cells. If we had a more heterogeneous population, this would be slightly different. This is just an example. So, well, we want to detect these highly variable genes that are candidates to be used for this clustering afterwards, or they are informing us about the drivers of heterogeneity. How do we do it? The way we do it is to look at this biological cell-to-cell heterogeneity component. So we'll have this variance decomposition here, and we can estimate each of these quantities. However, if we just get a point estimate, we're ignoring the uncertainty about that. So we want to have some kind of probabilistic measure of the information we have to decide that this gene is highly variable or not. So if we have a given threshold of variance, for example, we can say, I will say that it's a highly variable gene if the variability, the biological variability is at least 70% of the total variability that we observe, then we'll classify as a highly variable gene those genes where this proportion is higher with a high probability. So it's higher than 70%, but we also are very sure that it's very high, it's higher than 70%. So this sigma I is just taking the previous expression, and we took the third term divided by the total, and this is just simplified. Okay. Now, because that variance decomposition was like cell specific, we have the phi J's and the SJs. Here we only take one kind of like typical cell as a reference by taking the median of all these values. For the lowly variable genes, we do something very similar. So we look at the proportion of variability that is explained by these biological cell-to-cell heterogeneity components, and we want this component to be very low. For example, less than 20% of the biological variability is due to a cell-to-cell heterogeneity component, and then we want the probability of this event being very high. So this probability is telling us how sure we are that this event is true. Well, we have four kinds of thresholds here. So we have these variance thresholds, and we also have these evidence thresholds. So we need to have a threshold for deciding how much variability is biologically meaningful for you to define as a highly variable gene, but also we need to define a cutoff in terms of the probability. So the way we control this, so the variance thresholds, in general, you can fix them a priori. For example, you can say, well, I will be interested in 90% of the total variability. However, the evidence threshold is something more technical. To control these thresholds, we use a trade-off between false positive and false negative rates. So if we have a probability threshold that is very high, we'll tend to have very low false positives, because we require like this probability to very, very high to define it as a highly variable gene or lowly variable gene. But then we'll have lots of genes that we don't really characterize as highly variable genes because we are asking too high probability. So the false negative will increase. So there is a trade-off. So by looking at these two, so we turn this expression. Basically, this is just an indicator of whether the probability was higher than the threshold. So the denominator here will be just the number of genes that we classify as highly variable. And then here, what we have in the numerator is the probability or the predicted probability of not being highly variable. So it's 1 minus the probability that we are using for the classification. And to control these two, well, we can run a search for different values of these thresholds. We can compute the specs false positive and false negative rates, and then, for example, say, I would like to have the false positive and false negative at 10%, and that will give us specific thresholds for running the test. So now we have quite a complex model. One question is how we actually estimate this model. What is what we are doing internally. So we are doing Bayesian inference, so just a quick revision. This is Bayes' theorem. So if we have a quantity of interest, in this case gamma, we want the posterior distribution of these quantities of interest given the observed data, given what we saw. And the way to obtain this is to look at the likelihood. So this will be the distribution of your data given the parameter value combined with some prior information or prior density. And this is what we have here. So in our case, which prior we use? So we use a parametric prior for all the model parameters that we have. We share the priors. So in this case, we are not really saying that one gene is more highly variable or less highly variable a priori. We use exactly the same prior. However, we have done quite a lot of simulations, and we observe that these values are not too critical, the hyperparameter values, because in general, we have lots of cells. So the data actually wins in terms of driving the inference. So because we have this very complex model, we have lots of parameter values, and then therefore getting this expression in an analytical form is not possible due to this very high dimensional integral that we have in the denominator. Therefore, we go to numerical methods. So we use Markov chain Monte Carlo to do this. Before doing the Markov chain Monte Carlo methods, one thing that we do is to integrate out those ρij's that I had before, so those parameters controlling the heterogeneity. Because otherwise, the sampler gets there are too many parameters that need to be sampled from, and then the running time will be very costly, and the chain will not mix very well. And the algorithm that we use is an adaptive Metropolis Hastings within Gibbs algorithm, and I will go a little bit in detail with this. So for those who are not familiar with Gibbs Sampler, what is Gibbs Sampler. So basically, imagine you have a very high dimensional posterior, like parameter set, and you want to generate samples from this posterior. Instead of trying to aim to sample from the whole posterior, we can divide this in small chunks and sample from those chunks. Okay? And the way that is done is by building these full conditionals that will be the posterior distribution of one parameter given the values of all the other parameters and the observed data. And sequentially, we will start sampling from each of those parameters at each iteration. So for example, we will start with one initial value. At the first iteration, we will update the first, like gamma 1, from its full conditional, given the current values that I have of all the other parameters and the observed data. Then for updating the second parameter, I will look at the full conditional, but instead of using the value that I had at the previous iteration, I will have the value that I sampled just now, and the other values, because I haven't sampled them yet, I will use the older, and so on. If we repeat this a large number of times, the sample that we will obtain will resemble a sample that comes from the distribution that we will targeting. So in our case, the full conditionals of parameters of the same type, what I mean with the same type, for example, all the mu i's or all the sj's, they factorize. Why it is important is because this will allow us to do these updates, all of them simultaneously. So I will not sample mu I and the mu 2 and the mu 3 separately. I can sample all of them together because the full conditionals factorize. So the sample from mu 2 doesn't depend on mu 1. So I can just do them in parallel. And if I want, I could parallelize this in multiple processors. However, most of these full conditionals do not have a closed form, so I cannot really do a straight like a simulation from R, like using R norm or normal distributions that we know. We need to go to more specialized samplers. In particular, we use Metropolis hastings. So Metropolis hastings is an indirect way to generating draws from a specific target distribution. So you have a distribution that you don't really know how to sample from. What you will do is to sample from another thing that you know how to do it, and then given some quantity of how good or bad was this candidate value, you will accept it or reject it. So what we do is to sample from a uniform distribution and then generate some candidates from some kind of transition probability. You can interpret this as a transition probability, and then calculate some acceptance rate, which depends here. If you look at here, this is the posterior distribution of the parameter that I had the current value versus the new or the candidate value. So if the new value, the candidate value, is better, so this posterior probability is higher, I will tend to accept it. If this new value is worse, so this value is smaller, I will tend to reject that value. Again, if I sample long enough, I will get a sample from the distribution that I was targeting. Now, one big issue is that we need to know this thing. We need to know this candidate or jumping distribution where we are sampled from. A very common choice is to use a Gaussian random walk. So what will be this? Just to take that the new value or the candidate value will be drawn from a normal distribution centered at the current value with some variability. Now, the value of this variability parameter will affect quite a lot how many values we accept or will reject. So if we are sampling, for example, with very small value here for this variability parameter, all the draws will be very, very close to the current bound. So this quantity here will be, these two will be very close and will be close to 1. So we will tend to accept all the values. Whereas if omega square is too large, we are sample values that are too far apart. And then these two will be very different. And we might end up projecting too many values, so our chain doesn't move very efficiently. Therefore, one way to deal with this is to use an adaptive method. So every, for example, 50 iterations, you can stop the chain and look, well, how many times I did accept or reject the values that I was sampling from. If I accepted too much, well, maybe I need to increase a little bit this variability parameter. If I accepted too little, I need to decrease this variability parameter. overall, we are targeting that the acceptance probability is around 0.4, something between 0.2, 0.5 should be in an accepted range. And if we use these quantities that we increase or decrease are very small, or so they diminish on time, we'll have that this variance will kind of stabilize. So some final remarks. I'm not sure if I was too quick or was okay in time. So single-cell RNA-seq is a novel technique that will give us new insights about biological heterogeneity and what is going on at a more molecular level. However, analyzing this data is quite challenging, and in particular, we had issues with the quality control, the normalization, and the technical variability. And the tendency is to just think that methods that were developed for bulk RNA-seq can be directly used for single-cell RNA-seq. And that's an issue because for a bulk RNA-seq, we don't have all this technical variability going on, but much smaller than in single-cell RNA-seq. So therefore, ignoring that part will affect all the rest, okay? Because if we are not dealing with these two steps, and we just normalize the data without taking into account the noise will affect our normalization. So this will be an issue. So the approach that we presented today was instead use a simultaneous approach where we are taking care of the noise, the variability, all of them simultaneously taking into account intrinsic genes and technical spiking genes. So some extensions, well, people normally, what they are looking for this single-cell RNA-seq is not just to detect highly variable genes. Normally, the aim is to cluster your cells, to have very distinct populations of cells, and then to find the markers, kind of like a differential expression analysis, which genes are highly expressed here and lowly expressed in the other cell. So that's one of the extensions that we are working on now, but in such a way that we include the noise in between these stages. A second issue is scalability. So nowadays, sequencing is getting cheaper and cheaper, and we are sequencing more and more cells. So a few years ago, people will only sequence like 96 cells that go in a plate. Now there is a new technique called drop-seq, in which they basically put cells within some tiny drop, and they can do thousands and thousands of cells. So estimating these kind of models will become more complex, and the other complexity is we will have no spikes. So currently, they are not using spikes. So we will not have a way to estimate the noise. We'll need to find some different way to do it. And just a reminder, after the lunch, we will have a practical session. All the materials are uploaded in this GitHub tutorial. So please do download the files beforehand in case the Wi-Fi is a bit slow and we cannot download them all simultaneously. And just some acknowledgements. So people in John Marioni's lab, which we are in the EBI, but recently moved to cancer research as well. So we are in between the two institutes and the biostatistics unit. And in particular to Niels, who is sitting here and will be helping during the practical session today. Any questions or?